Hi, I'm Connie Lee, and I'm here today with Dr. Amy Akers, who is the Chief Scientific Officer of Angioma Alliance. And I'm going to be talking with her a little bit today about her role as both as a scientist and as a member of the Angioma Alliance team. Amy, can you tell me a little bit about how you became interested in science and specifically in cavernous angioma research?
Hi, Connie. Yeah, sure, absolutely. I guess it all goes back to being a little kid and having just a really intense interest in science and nature and animals. I always loved bugs. For example, when I was a little kid, I had butterfly collections and we'd pin them on the walls. You get the pins, you hang them up, and we still have butterflies in our bathroom. And I'm fascinated by them, fascinated by science in general. As an undergraduate, I really thought I would pursue a medical career. I thought medicine was the path for me. I started volunteering in a hospital and in a nursing home, and really I found out that wasn't right for me. But what was right for me, I did a research experience at the vet school at Cornell where I did my undergrad, and it really changed my plan. It opened up so many doors for me, and I just became so excited by the research portion of science that I decided to pursue a PhD in human genetics, where I went to Duke University and joined the university program of genetics and genomics there. And I really wanted to work in a genetics lab where I could have some human disease experience. So kind of marry that medical school desire with basic science. So while I was at Duke, I joined Doug Marchuk's lab. Doug's an expert geneticist and studies mouse models of a variety of vascular and cardiovascular diseases, one of which is cavernous angioma. So I started working on the CCM projects there, and my project was focused on understanding the genetic mechanisms for lesion genesis. So what is happening at a genetic level within the endothelial cells, the blood vessel cells of those lesions that's causing them to form. Importantly, the project required using CCM tissue from patients. And this was an issue because my boss, my advisor, was not a surgeon, so we didn't have surgical tissue around. It's a very, very hard thing to access. But fortunately, Angioma Alliance runs a wonderful DNA and tissue bank and had tissue that we could use. We were able to access tissue that way, and I completed my PhD and had some really cool findings. So during my days in the lab, I got to know Connie very well because of the need for tissue, and of course, she was running the CCM scientific meeting at that time. So I really gained a true appreciation for the power of advocacy organizations like Angioma Alliance. And when I completed my PhD work, I was ready to move out of the lab. Still wanted to stay in science, but not do a traditional scientific career path. And I jumped on the opportunity to join the Angioma Alliance team, and that was more than 7.5 years ago now. And it's been an absolutely wonderful opportunity and experience.
Wonderful. What is it about your position that brings you the most fulfillment?
The position is really cool. The intellectual stimulation and opportunity to learn are just absolutely fantastic. No 2 days are ever the same. For example, I'm talking to you today, which is really fun. And it's very satisfying to work on a variety of different projects. For example, my work does still include some basic research to which I'm accustomed, but also I've recently had the opportunity to work with the New Mexico Legislative Office of Senator Udall to develop and introduce the CCM-CARE bill. This is a piece of legislation intended to increase awareness and funding for CCM research and drug development, and to support the development of our CCM clinical centers of excellence. Another very different project I worked on recently is to develop the clinical care guidelines for CCM. This is a project I'm very proud of, and within the next month or so, a comprehensive guideline for clinical management of CCM will be published in a peer-reviewed journal and also on Angioma Alliance's website. I'm the lead author of this paper, and I'm really grateful for other significant contributions from the clinical members of our scientific advisory board and experts to really bring this paper to fruition, and I think it'll have a huge impact on the patient community. And at the end of the day, that's what it's all about. It is super rewarding to be the most rewarding part of this position is knowing that all of the work I do has a direct impact on the CCM patient community. Without a doubt, my work and the work of Angioma Alliance has and will continue to push the frontier of CCM research and bring us closer and closer to clinical trials and real non-invasive drug treatments.
Yeah. Yes, absolutely.
Yeah.
You are no longer in a lab, as you said, doing traditional bench science, but
Right.
Do you consider yourself to still be doing research, medical research? ways.
Sure. Absolutely. I'm the lead investigator on 2 research studies right now, one of which is our biobank that I mentioned before. So the biobank is actually a study where we collect DNA, we collect tissue, we collect medical records and imaging records, and we can use that for research, our own research within Angioma Alliance, or we can share those resources in a de-identified manner with other laboratories. So very much involved in that recruiting patients and the research end of the biobank project. And then also, I'm a lead investigator for one of the sites for the Brain Vascular Malformations Consortium. So this is a project that actually studies 3 different vascular illnesses. I'll just talk about the CCM portion of it. There's 4 sites: Angioma Alliance, University of New Mexico, University of California, San Francisco, and the University of New Mexico. And essentially what we're doing, it's a genetic study to collect information on people. So we're collecting samples, DNA samples, and then also clinical records to look at the variability of this illness. CCM is highly variable. Even within families where people have exactly the same inherited mutation, you would think they'd all show the same disease spectrum, but they don't. There's some people who basically are symptom-free and others who unfortunately are not. So we're trying to figure out what other effects are modifying the symptomology of CCM. So those are 2 very obvious examples, clear examples that I am definitely still doing research. But kind of in a broader sense, research I don't do myself, but I definitely am facilitating research on a very, very regular basis. I'm involved in supporting the research through collaboration building, really, bringing people together, the CCM meeting, bringing a new industry involvement into the Angioma Alliance fold, so to speak. BioAxone is a great example of that. They're a small biopharmaceutical company out of Cambridge, Mass, who is studying Rho kinase inhibitors. And Rho kinase inhibitors are probably going to be one of the big drugs we're looking at, or big drug mechanisms we're looking at for clinical trials down the road. And they were studying them in a very, very different platform. And I said, hey, I reached out to them and I said, hey, CCM science is really interesting. There might be some cross-fertilization here. And, they came to our meetings and they've now started these public-private partnerships with some of our academic researchers to push forward a drug that they're working on. So, that's not direct research, but it's still a very, very important thing. Bringing people together. We also have great relationships with folks at NIH, and we're starting to work on a new relationship with the FDA as we're preparing for clinical trials. So I think relationship building is a huge part of what we do. And Angioma Alliance has a lot of other resources that can help support research, even down to study design consultation for clinical trials, talking to investigators about the best way to design trials and what is the best outcomes for our patients, and will patients actually participate in a trial that requires this, and that. So those are conversations that we're involved in. We provide samples from the biobank. Like I said, study recruitment is huge for us. Our patient registry is a fantastic tool that we have. Angioma.org/registry is a fantastic tool that we have for study recruitment. We can reach out to people who are interested in being involved in research studies and really increase recruitment for studies, both basic research studies that are happening now and clinical trials down the road. It's just a really nice connector between the patient community and the research community who doesn't have that direct contact. And then even now we're helping folks with preliminary data, sending out surveys to our members, asking a variety of questions about maybe drugs they've taken or experiences they've had and how that pertains to their symptomology. And that's really been helpful for preliminary data for some of our basic researchers to obtain funding for new studies. So, it's all over the place and it's a lot of fun and really interesting.
Yeah, thank you. We're going to have a second little chat later on about the specific medications that are under development and the Angioma Alliance role in bringing them to patients, as well as some of the obstacles and how we're trying to overcome them. But I'm glad we had this bit of an introduction to you first, and I look forward to talking to you in the future.
Absolutely. Fantastic. Thanks, Connie. It's great to see you.
Bye-bye.
Bye.